The title of this presentation is Accelerated Molecular Structure Discovery with Variational Quantum Eigensolvers for COVID-19 and CellTalkDB Receptors. To begin with a presentation overview, we'll be motivating the utility of molecular docking workflows in the fields of enzyme discovery, directed evolution of enzymes, machine learning, and deep learning for being able to discuss how a wide variety of molecular structures have connections with other bioinformatic workflows, and how very much, if we want to do some discovery or more exploratory data analysis and searching through different possibilities for ways that data from different types of experiments or different areas of biotechnology can be related, we can look to this molecular docking workflows that we're going to be describing. And in doing this, we'll have to introduce objects to bridge computational chemistry and quantum computing from objects that we'll be defining and discussing more, like the electronic structure Hamiltonian, or the 1-point and 2-point electron correlators and the like. And we'll study as use cases the COVID-19 receptor and CellTalkDB sequences, which will allow us to determine what are the differences and different characteristics of the algorithms for quantum computation and classical computation alike that can not only be addressed from these types of algorithmic workflows, but also what are the types of difficulties and intrinsic characteristics of each types of these algorithms. And this is to say that there are many bioinformatic algorithms and pipelines which share connections with molecular docking workflows. For one, we can see that I've listed a couple of videos that were previously given on the channel that relate to some form of structure prediction, in which I looked at ways to approximate the binding affinity for different types of compounds and to be able to compare an in silico result that's generated from simulations to some type of experimental result that's already known. On the other hand, we can look at the probability of encountering uncertainty in sequencing or in sequence assembly with de novo graph assembly, in which from a FASTA file, which can contain hundreds of thousands of different sequences, as well as conserved regions and different types of characteristics of the motifs, we can look to visualize the graphical structure in which there can be an uncertainty in the algorithm on certain base pairs that it predicts for different sequences that are contained in the FASTA file. So this allows us to make more sense of a bioinformatics pipeline or workflow that produces hundreds of thousands of data points from which we can look to characterize the differences between several sequences, or even hundreds or thousands at a time. And this is not to mention that if we want to make some other type of connection between what we can view and visualize in a de novo graph assembly with Bandage, we can also look to make additional changes to open source workflows for, say, reverse translating amino acid sequences or sequences to different types of formats, so that we can form more interpretations rather than just a transcriptomics or genomics workflow. And we can look to form some type of interpretation, say, of the epigenome or other characteristics that has some biological, different biological characteristics. And on more of the cellular level, we can even look to describe cell annotation and clustering and heat maps with an open source platform that's provided by the Broad Institute, which allows us to input very much the same type of FASTA files that we use as input for the de novo graph assembly, which is a different type of bioinformatic pipeline, because the cell annotation, clustering, and the heat maps in Morpheus, which are functionalities that are provided in Morpheus, is completely different from a more of a visualization type of application that's provided in Bandage. And finally, in the last item, we can also look to the transcriptome assembly, which is another bioinformatics pipeline that's provided by developers at the Broad Institute, in which you can look to provide as FASTA files as input to the transcriptome assembly, so that the transcriptome assembly can assemble some collection of sequences, which we can call the transcriptome, which we can also look to quantify the relative abundance of with another software package, Callisto, from the Patra Lab at Caltech. So there are many different types of applications that can relate to molecular docking frameworks and workflows that we're going to be drawing upon in this short talk. And in the next slide, we can also observe that there are preloaded data sets for cell annotation, which can unveil mechanisms by which receptors of different disease propagate infection to host organisms. And this is very much related to the fact that when we're looking to run a molecular docking simulation, there can already be a lot of information available, such as a preloaded data set for cancer or other types of cancers that develop in other types of the body, and how having this information about how previous cases of the disease receptor have interacted with components or parts of the human body and have propagated an infection can be helpful for looking at different consequences of these algorithms, as well as trying to make a broader implication about what types of ways a disease could spread depending upon the receptor of interest and whether it's associated with the disease. And this is to say that we don't just have to rely on classical methods of computation for being able to look at ways in which receptors and ligands can bind and interact, as well as how other molecular vesicles can interact with the receptor of interest. We can look at approximating the behavior of nonlinearities of partial differential equations, which can relate to complicated networks, genomics networks, and genetic networks, in which the presence of different types of chemicals, such as RNA polymerase and other quantities, which are available in the body in interactions with DNA and RNA, which can help us have an idea about how we can generalize and apply our workflows to instances in the cloud for being able to further automate machine learning and deep learning workflows. And from the bioinformatic pipeline, which we had described earlier, which was relating to visualizing de novo assembly graphs, when we make use of a FASTA file as input, which can contain on the order of hundreds of thousands of sequences, we can see that once we import the FASTA file into the bandage graph application and look at the resulting graphs, there can be hundreds of thousands of nodes and edges, as well as, in this case, being around 63 million nodes, which is definitely a significant increase in the complexity from just the sequences that are contained in the FASTA file themselves, because there can exist many other different types of ways in which these sequences can interact with one another and form characteristics and traits of complicated networks that we would like to experimentally validate or also be able to characterize in silico. And in terms of connections with even other areas of physics, such as biological physics and quantum physics, we can observe that the spectra of potentially large random matrices can be formulated in terms of properties of biological systems and open quantum systems, which are characterized by the fact that these systems are able to exhibit, even though one system is a biological system and the other system is an open quantum system, the fact that they can have some ensemble type behavior, which can describe the growth of bacteria or films, which is very much a a set of related topics that Google Quantum AI is looking to explore in its more recent near-term quantum hardware, which is depicted on the left side. And as given on the right side, there can also be very much an important area of research and important area of future attention to look at how we can stabilize entangled states, as what is discussed in this reference from 2012, which can give us an idea about how we can accurately time evolve different highly entangled quantum states and be able to generate meaningful measurements and readout from these states. And depending upon the particular type of molecular docking workflow that we're looking at, to make a connection with quantum computing with. In a previous presentation on the channel, I gave an idea about how one can look at different publicly available information from the PDB repository, the Protein Data Bank repository, and how having this type of information can be used to approximate the binding affinity of CellTalkDB compounds with AutoDocVina. So it's very helpful and a very encouraging step in being able to demonstrate that this molecular docking workflow can be of interest for making connections with other types of algorithms, in which from a paper that I'll introduce formally in a reference, there was a study on the COVID-19 receptor in which the authors from this archive preprint were able to conclude that from the 92 available crystal structures that they use as training data for their deep learning algorithm, that they were able to look at a distribution of protein binding or pockets along the surface of the COVID receptor, and they were able to anticipate how different conformations in different molecular vesicles or even receptors could interact with these binding pockets, which was an output for their deep learning algorithm. To introduce a second paper reference, this is to also mention that there are other possible databases for looking at SARS-CoV-2 in drug candidates and being able to effectively tackle how this receptor propagates its genetic material throughout the host organism after infection. And this is to say that it's related to protease, to the production of protease in the coronavirus, which is related to some types of similarities between how some types of diseases propagate, as we had just mentioned, their genetic information and continue to infect the host organism after having an interaction event with a different part of the host organism at which the infection initially occurs. And from the second paper reference, by making use of AutoDocVina, which was the same type of open framework that I had used and discussed in a previous video, the authors of this paper can look at different conformations and different orientations of a ligand of interest, which is a much smaller compound in comparison to the receptor that's interacting with. And they were able to generate these types of predictions from running AutoDocVina and then looking at systematically from however many conformations are produced in the PDB-QT output format, how from this PDB-QT output, they can look at using either the Python molecule view or other types of open source molecular platforms, how or what would be the expected modes of interaction between the receptor and the ligand, and how this can relate to protease of the coronavirus. And on a similar note, the type of open source molecular docking workflow that I applied for CellTalkDB sequences was given from this website, which is maintained by scientists at the Chinese Academy of Sciences. And they basically present and they give an overview of a collection of manually curated sequences in which they provide approximately 3,400 ligand receptor pairs, but they also provide a different or additional information on the ligands, receptors, as well as also ligand and receptor pairs for mouse organisms. So this can be really helpful because it can help demonstrate whether there be any predicted similarities in the conformations that are produced from our in silico molecular docking experiments, and whether being able to run this characterization in silico in the fact that not having any experiments to do initially allows us to have some better type of idea about which compounds would interact optimally with a given receptor and how this would help us be able to look at the best candidates from hundreds of thousands of possible candidates, or even potentially even more than that. And on the CellTalkDB GitHub, they give us an overview of how their workflow and the collection of ligands and receptors that they gather for the human and mouse are related. And this is given by the fact that in the very first step of their data collection stage, they look at protein-protein interactions, which they abbreviate as PPIs. And they also make use of annotated information from several different open source repositories, such as the NCBI Genome or UniProt KB, and how making use of this information allows them to gather enough information so that they can add on additional information in the PDB repository. Because the data collection that they have in the beginning step doesn't exactly have all of the information or the information that's needed in the format that's desirable for running a molecular docking workflow in a simulation. But rather than previous applications of molecular docking workflows and CellTalkDB sequences and protease inhibition, we would like to think about whether there's any way we can make a connection with quantum algorithms, because there's a lot of industry and a lot of focus on quantum algorithms in looking at potential connections with electronic structure and quantum chemistry and computational chemistry, and determining whether we can theoretically predict any desirable properties of different chemicals and materials. So this is just to say that the structure of the COVID-19 receptor, which I mentioned in a previous slide and which I've reproduced here, it has a certain distribution of binding pockets which can be obtained or gleaned from the 92 possible available crystal structures. And then we also want to think about how this can be connected and related to the CellTalkDB sequences, which have this data collection stage and also a type of a data cleaning stage for providing us information from the PDB files of the ligands and receptors, and how leveraging these two types of perspectives can be used or can be reinterpreted and can be studied further in a quantum computing framework. And this is very much relating to the reference, which I'm going to be discussing some of the figures from the paper from, so that we can further connect and make use of insights in one area and transfer it to quantum computing and vice versa. Maybe start with some type of interpretation from quantum computing and which can relate to the quantum structure and the electronic structure from a quantum perspective, and be able to also look at this from a more classical molecular dynamics machine learning perspective. So this is just to say, why would we want to consider a variational quantum eigensolver, which is one particular type of quantum algorithm of interest, which relates to the variational quantum algorithm, which is also abbreviated similarly as VQA. And this is because there can be many potential applications for quantum circuits that someone can design in time evolve forwards to generate some type of measurement, which can be used in the VQA framework to determine whether there can be any quantum circuit or classes of quantum circuits, which can provide us readout that's informative for approximating the binding affinity or other properties of the electronic structure of different materials. And this is just given by several different types of work, such as a conference presentation that I gave at the APS April 2022 virtual meeting. Also, some type of work that I'd further discussed in terms of shallow wave propagation at a Dartmouth seminar virtually, and also a couple of other virtual presentations that I have on the channel, including looking at a generalization of the variational quantum eigensolver, which was related to the APS March 2023 meeting, as well as two other presentations on the channel in which I looked at a connection between physics-informed neural networks and being able to train a physics-informed neural network from the output that's generated from a quantum algorithm, and also looking at how to apply the framework for solving PDEs in terms of a biological transport perspective, in which we can look at the time evolution of different trajectories of cells or other vesicles that are contained, or molecular vesicles that are in the body, and how we can look at to reconstruct different types of trajectories of such particles with information from a quantum algorithm. So this is just to say that there are many connections, and there will continue to be many connections that will also be developed in terms of the variational quantum algorithm, the variational quantum eigensolver, physics-informed neural networks, deep neural networks, which can give us a framework that we can situate ourselves within to begin discussing from more of a quantum perspective, the molecular docking workflows. And now I'm going to, go back to the general VQE background, which is given by this reference. So I'm going to discuss how we can look at different ways in which the variational quantum eigensolver has already been used, and how we can look at making use of this framework and this background to look at the electronic structure of new compounds of interest. Okay, and so this is the workflow diagram, which is given by figure 1 from the earlier reference that we had mentioned for quantum computing for drug discovery, in which they describe an iterative optimization loop, which is very much characteristic of many different types of workflows that are given in biotechnology, whether it's more in instances or in working environments or in academia or in industry, in which many people start with looking at virtual types of characteristics or properties that they can observe about a compound of interest without having to carry out any types of experiments initially, and determining whether they can look at or systematically rank or predict from more of a first principles perspective differences in the electronic structure of compounds, which would interact either more optimally or less optimally with different types of receptors. So this is just to say that from the first item that's given in their workflow for the drug backbone discovery, you can perform a lead optimization, which is based on many different types of possible characteristics, such as we can see, you can look at ligands, ligand base, as we have been mentioning and discussing more, or we can also look at databases and make connections with databases and scoring functions, much like a scoring function which is used in Autodoc Vina and other types of molecular docking frameworks, as well as looking for a drug structure selection, namely determining whether there are any regions of the surface of the receptor that would either interact optimally or could actually harm an organism if the interaction is based upon a different binding pocket on the surface of the receptor. But then we can also look forward to more connections between quantum mechanics and molecular dynamics simulations in the bottom part of the workflow, because that's where more of the quantum mechanical computations can come into mind, and which can then trickle down to experimental assays, which more experimentally minded people can be interested in exploring and confirming whether the type of assay that they can develop from the lab can generate the type of prediction that's generated theoretically. And this can go on and on and on, with even connections towards running the computations on quantum circuits and even near-term hardware for determining whether this part of the workflow can also be, which is revolving around computing reduced density matrices, can even have a positive and far-reaching effect in terms of development for experimental assays. And such workflows relate to those of enzyme discovery pipelines, which is given such as in the paper from the Arnold lab, in which from many papers over decades that have been talking about this type of directed enzyme evolution or directed discovery of enzymes. In this particular reference, which is more recent from a couple of years ago, which was in 2022 and appeared in 2024, is talking about machine learning. So it's just demonstrating that there can even be connections between quantum computing and quantum machine learning in determining and in realizing applications of these workflows that could speed up enzyme reactions. And so more generally, enzyme discovery pipelines have also applications in the following industries for designing enzymes with high catalytic activity, such as agriculture or drug discovery, as we have been mentioning more on the drug discovery side, but there can also be applications more on the agriculture side in being able to determine how certain chemicals can react optimally for guaranteeing the safety of crops in extreme weather conditions. And from the quantum computing reference that we have been mentioning, one topic that's of central interest to the authors of this paper is approximating the Gibbs free energy, which is appearing as delta G in the molecular dynamics literature with Hartree-Fock and other types of quantum algorithms, and how this can be related to VQE or density functional theory. So we basically want to think about whether Hartree-Fock and the electronic structure Hamiltonian and other related objects that we can define from the Hamiltonian can be leveraged to generate thermodynamic interpretations of how well different conformations interact with a receptor of interest. And from the variational quantum algorithm that I had mentioned earlier that I've experienced in running and compiling quantum circuits on, we can already see immediately that components of the workflow that are shown in the previous slide from the reference that we have been discussing share several connections with the variational quantum algorithm, the VQA workflow. In the VQA workflow, we were looking at the nonlinearity space for some partial differential equation of interest that we wanted to study further. And then we impose requirements on the ansatz, which is the initial state of the solution, much like how someone can look at imposing requirements on the initial state of a molecular structure that would guarantee its stability and being able to also satisfy other desirable thermodynamic properties once a binding event between a ligand and receptor has occurred. And this is just also demonstrating that once we pass from the nonlinearity space in the initial state of the solution that we would like to require for the PD through the ansatz, we can look at formulating a cost function ground state space, which is very much relating to the types of terms from the electronic structure Hamiltonian and Hartree-Fock frameworks, which can help us determine which type of thermodynamic interactions are favored in the molecular structure, which would overall be able to generate or to guarantee the stability of the structure. And finally, the quantum circuit computation comes into play, much like the workflow that we had demonstrated from the quantum computing reference, with which had mentioned connections for further downstream processes with experimental assay design, because we can look to compile our results on a quantum circuit with Circ, which is the open source framework provided by Google Quantum AI, which allows us to reconstruct different portions of the approximation for the binding affinity, or other types of experimental measures for the catalytic activity of a compound of interest, which we can then help use to leverage to make some type of thermodynamic interpretation about how effective different types of compounds or molecular structures would be for guaranteeing that some desirable type of reaction occurs. And now that we have been making connections between molecular docking, machine learning, deep learning, and variational quantum algorithms and variational quantum eigensolvers, we can actually demonstrate an example of a quantum circuit that's provided from the reference that we have been describing some figures from, which can help us get an idea about different types of aspects of the quantum computation that come into play through the circuits. In particular, what we need to keep in mind when we're reading a quantum circuit is that there can be different types of registers, which are given by the Q0 and the Q0, which represent 2 qubits, which we initialize in the leftmost part of the quantum circuit, as well as the fact that we can have 2 rotation gates, which can then be connected, in which the qubits Q0 and Q1 can interact with one another and become entangled, after which we can provide the quantum circuit has 2 other gates in which we can perform some type of measurement or some type of rotation on the qubits before obtaining a measurement gates from the hat of the poly Z operation. And the last 2 gates are given in the quantum circuit to the rightmost of the quantum circuit diagram. So this is demonstrating that we can look to make connections between circuit design and how this influences the type of quantum computation that we would perform and what type of molecular-based information we can get from the quantum circuits that we can reconstruct some measurement from. And there's general VQE background, which I'm going to just very briefly mention throughout the presentation for completeness so that we can continue to describe how different parts of these workflows can all be related together. And this is just one particular reference on the variational quantum algorithm eigensolver, in which it provides a review of the methods and best practices. So I'm sure that there are several other types of references that give a similar notion or impression of what the variational quantum eigensolver is and how we can apply VQE, but this is just the one that I just chose out of convenience. And the general VQE background is based on a similar type of workflow diagram, which we had reproduced from a previous reference, because as we have been mentioning for Hartree-Fock or electronic structure Hamiltonian, there can be some type of Hamiltonian representation as they given here, which is given by the electronic structure Hamiltonian, which is comprised of a Hamiltonian with 2 terms, one 2-point interaction for P and Q, because we're looking at the product of the annihilation and creation operators at different sets of the square lattice, about which we can have some interaction, as well as 1/2 times a summation for a 4-point interaction, which is also related to a creation or a product of annihilation and creation operations. And we can look to perform an encoding of this Hamiltonian. So in a way, we're taking the Hamiltonian for the electronic structure and mapping it into a different type of Hamiltonian and being able to look at ways that we can perform this computation for the time evolution to determine qualities and characteristics of the molecular structure from the quantum circuit that we're time evolving. So this is just given that from the fact that Hartree-Fock can be used as one initialization scheme for our molecular docking simulation that we're carrying out on a quantum circuit. And then, as in the variational quantum algorithm from Lubash et al., which I had reproduced, which I had reproduced the workflow of that I presented on in the APS March and April meetings from 2022 and 2023, we can see that there are ways in which we can look at a sequence of gates which can involve C-naught gates or other types of rotation gates and quantum Fourier transformation gates, so that we can have some state preparation of the ansatz, which will position our simulation for probing and studying the electronic structure in terms of a favorable initial state, so that we can generate some beneficial and informative readout from the quantum circuit. And then we can also look at additional other steps which are given by 2B, 2C, and 2D, which are pretty much relating to what we have been mentioning and alluding to in previous slides, in which we can look to generate some meaningful type of measurements from polystrings or other types of states that are prepared on the quantum computer. And then we can look to distribute some type of hat poly Z gates, which are going to allow us to reconstruct the measurement for the from the quantum circuit, which will then have some thermodynamic or molecular interpretation. And this is also another picture of a quantum circuit, which is provided in the VQE background paper that we had mentioned 2 slides ago. And it's very much demonstrating how the quantum Fourier transformation gate and the Hadamard gates play very significant roles in being able to estimate different phases of quantum matter and being able to estimate the quantum phases. So this is very much related to other types of variational quantum algorithms or quantum algorithms in general, which are relating to quantum amplitude and summation, or even looking at being able to solve specific narrow classes of partial differential equations with a quantum singular value transformation. So this is just to say that the types of gates and components and the gates that we're using, which can be connected with molecular docking simulations, have very much been used in different types of algorithms, and how looking at it from the algorithmic perspective of one class of algorithms can help us have an idea about how these ideas and notions would transfer over to other classes of algorithms for which we maybe haven't ran as many numerical experiments on yet. And the VQE background will be later be further built up further upon to demonstrate how the electronic structure of the Hamiltonians permits for quantum classical benchmarking. And what we basically want to have an idea about when we're looking at comparing quantum and classical methods of computation is whether we can either realize quantum advantage from our quantum algorithm by being able to generate with high fidelity and with high probability an output from the quantum algorithm, which is in very strong agreement with the classical algorithm. And furthermore, whether there would be any type of quadratic or exponential speedup with such a class of algorithms, and whether there would be any good theoretical reasons for why the quantum algorithm is able to generate a very similar answer as the classical algorithm does in a polynomial or exponential speedup in time, or quadratic speedup. So there can exist many different types of speedups for the algorithms, and unfortunately, sometimes with the current state of research, and they're not being exactly clear how one can generate, or fabricate a large scale quantum device that's on the order of millions or hundreds of thousands of logical qubits. It remains uncertain whether some of these algorithms would actually generate or deliver on as much promise as many people in the industry have been saying that they have. So it's still very much an open question of interest and an open question of research to determine whether there be any types of algorithms which could help in providing a speed up while also being able to maintain fidelity to a solution that's already been classically computed. And we're going to be introducing more of these quantities later on for a reference on the electronic structure Hamiltonian, which I'm going to give. But I just wanted to very much just reproduce them earlier in the presentation because it can be something that the viewer can keep in the back of his or her mind, because we can have expressions for the electronic structure Hamiltonian, just like how we had expressions for the very same Hamiltonian in a previous workflow slide, and being able to map this Hamiltonian into another basis, which can then help us determine whether there are any thermodynamic interpretations, such as from the 1-point electron correlator or the 2-point electron correlator, as well as using the UCCSD ansatz, which is given by an ansatz that's parameterized in some minimum angle theta min, that is mentioned further in their paper, and how this ansatz is particularly informative and helpful in their purposes for running the molecular docking simulation. And as we have been mentioning and reiterating a couple times, connections with other works on VQAs has no mention of quantum chemistry and electronic structure computations. So this is very much why it would be helpful to determine whether we can make use of and leverage some type of PDE perspective, such as the variational quantum algorithms for nonlinear problems, which I had mentioned that I looked at earlier and ran simulations to approximate solutions for 15 PDEs in the framework of this paper, whether we can look at any other closely related classes of quantum circuits and whether this can be used and leveraged for a molecular docking perspective in a drug discovery workflow. And I just wanted to provide a little bit more background over a slide or two about the Lubash et al. Paper and how really this PDE perspective can directly be applied for generating time evolution on different classes of quantum circuits, and how this very much can demonstrate how a molecular docking workflow for electronic structure calculation for different compounds can pretty much be rephrased of and reinterpreted in terms of a quantum physics perspective. So this is related to the VQA, the variational quantum algorithm for nonlinear problems paper, because they formulate a cost function by expanding a norm in which they look at how they can transform a given partial differential equation of interest to a cost function, which informs how long the optimization procedure goes for, as well as the cost in terms of the cost of computation for an optimizer for different terms in the PDE. And this is to say that we time evolve a quantum representative solution of the PD solution. And then at each step of the optimization loop, before we time evolve the state on the quantum circuit and on the quantum computer further, we variationally modify the parameters in the quantum circuit insofar as to minimize the energy. And this is just to say that directly from their paper, they express the cost function in terms of a time evolved variational state, meaning they start with some initial state for the solution and then they time evolve forward. And then they provide some complicated initial looking expression in terms of the square of a variational parameter not minus 2 times the real part of some complicated quantum superposition, which is involving operations essentially on the zero state of the solution, as well as some constant term. And so having the cost function in the quantum circuit in mind, it relates to the same type of iterative optimization that we want to pursue and partake in when we're looking at ways to generate new insights which can help increase the speed at which experimental assays are developed. Because we have a backpropagation loop between the cost function and the quantum nonlinear processing unit, the CNPU, which is a portion of the circuit that's given in Lubash et al., which is relating to a bunch of input ports depending upon how many qubits you initialize and time evolve in the circuit, as well as a backpropagation loop 2, which is given by between the quantum nonlinear processing unit and the cost function, which is allowing us to run this variational quantum algorithm in polynomial time, because there are polynomial depth circuits in the quantum circuits that we're time evolving for the algorithm. So it's just very much relying on this type of framework and determining whether there are any types of quantum circuits, other classes of quantum circuits, which have polynomial runtime and which can be used to generate some type of illuminating information about the receptors, either for COVID-19 or other variants of the COVID-19 receptor, as well as looking at different connections between other diseases, whether it be a respiratory disease like COVID-19 or other diseases which can attack other parts of the body and infect other parts of the body through an initial replication stage, such as the groups of receptors which are given in the CellTalkDB repository maintained by those scientists, which I'd referred to earlier in the slide. And so in Lubash et al., as we had mentioned, they also provide a cost function for the inviscid Schrodinger's equation, the inviscid Burger's equation, rather than a cost function only for the Schrodinger equations, which raises the possibility of being able to use this algorithm or quantum circuits in a similar fashion, or that are designed in a similar way for other types of problems, like we've been mentioning, relating to the molecular structure and the molecular docking algorithms. And because there in other quantum computing workflows which study molecular docking and drug discovery, there are gates which are given in the quantum circuits which are related on diffusion operations. And we very much study nonlinearities from the variational quantum algorithm, which are related to diffusion operations and diffusive processes, which can relate to the process in which a molecular docking event occurs, in which after the catalytic activity has occurred, a diffusion gate or a diffusive process occurs, which stabilizes the receptor onto the ligand, in which results in a final conformation, exactly like the conformation that's predicted from the molecular docking workflow. So this is just demonstrating that the types of nonlinearities which we encounter in a partial differential equation can very much be related to other nonlinear processes and diffusive processes such as Brownian motion and other stochastic processes in the molecular docking workflows that we're looking to run and simulate different compounds on. And the quantum circuit architecture for the variational quantum algorithm basically relies upon the fact that we want to look at different types of connections that we can have from the quantum circuit, which we have on the left-hand side, which is given by figure 3 of the reference, in which we're talking about 2 qubits and we perform some rotation and entangling operation, and then another rotation operation on the qubits from which we generate measurements, to this type of circuit construction, which is given in Lubash et al., which is given by the fact that we initialize the qubits in the quantum circuit in the 0 state. We perform a Hadamard test in the topmost register of the quantum circuit, followed by other types of gates which are parameterized in the variational parameter lambda, as well as the fact that all of these qubits can be input ports to the quantum nonlinear processing unit, from which we can perform other Hadamard tests in the topmost register, as well as poly-Z rotations for generating the readout. And to perform time evolution on quantum circuits, besides the quantum circuit diagram, it remains for us to consider what is the initial state of the solution for the binding affinity approximation after each qubit is initialized in the 0 state. And relatedly, is there any trade-off between the number of qubits, i.e., one measure of complexity of the quantum circuit, and the fidelity of the binding affinity approximation to experimental values, if such values exist? And finally, with the third bullet point, which candidates could be examined within this framework to obtain preliminary results. So we're very much looking at the interplay between, in this quantum computing framework, for a different type of algorithm rather than a classical machine learning or deep learning algorithm. But with this types of quantum algorithms comes completely different considerations, because we have to look at how to maintain the fidelity of the solution when we start the time evolution, as well as how to maintain the fidelity of a different solution for a quantum representative state of the binding affinity or other electronic structure or other electronic properties of a compound or a material, and how we can continue being able to generate very high fidelity measurements and measurements with high probability that are accurate throughout all points of the time evolution. So now that we've been describing quantum circuits for different applications, and we've been looking to juxtapose how the types of gates appear in different quantum circuits, we can look at what the construction of the ansatz for the initial state of the solution would be when we're looking at a previous application of the variational quantum eigensolver and the variational quantum algorithm. And in this case, the ansatz is dependent upon quantum Fourier transformation gates that provide the following encouraging preliminary results. And this is to say that we need to generalize or we need to initialize the state before we're performing the time evolution, so that we can look at which type of ansatz for the initial state would perform favorably in the variational quantum algorithm and the variational quantum eigensolver. And it depends on CNOT gate, rotation gates, and Toffoli gates. And Toffoli gates have a particularly large overhead in making quantum circuits and in time evolving quantum circuits in comparison to other gates that we can make quantum circuits out of. So this is very much the type of consideration that we need to give in mind when we're looking at different quantum circuits, and what is the overhead with initializing qubits that we want to use, physical qubits and ansatz or ancillary qubits, which we want to use to encode a logical qubit, because even people at Google Quantum AI and other leaders in the industry, such as IBM and other people, which make use of different error metrics, such as quantum volume, for looking at how well a quantum circuit or a quantum device performs, also have drawn our attention to the fact that we need to potentially use on the order of thousands of physical qubits to encode a very pristine logical qubit. And after time evolving the quantum circuit for the first iteration, the optimization loop continues from the readout. And this is very much the same process that we want to continue and maintain in looking at a quantum algorithm for molecular drug discovery and molecular structure prediction, because we want to be able to further iterate and improve upon our approximation for the binding affinity for a compound of interest. And several compounds that have previously been examined with classical means of computation can be further examined within the quantum circuit approach. And this is just a reproduction of some compounds that I had looked at in a previous presentation, where I look at the CellTalkDB repository and I had ran homology models for several different types of compounds so that I can have some type of visualization and intuition about the structure of different compounds, which can then allow me to run simulations within certain search volumes for approximating the binding affinity. So this is just to show that because there are thousands and thousands of different diseases and different ways in which a virus can propagate its genetic information, that there can be different types of ways and different types of structures, as well as or in addition to several combinatorial ways in which a receptor can interact with a ligand, or other types of protein-protein interactions, which can be related to the wide variety of structures of hundreds of thousands, if not millions of structures for different viruses that are available. And from this collection of candidates, we can begin to make connections between the quantum circuit approach depending upon the complexity of each structure. From one of the structures, which I had just taken and isolated from the previous slide from the CellTalkDB database, we can see that there are different ways in which we can perform a rotation of this molecule, and how this would actually allow us to determine whether there are any types of symmetries or rotational symmetry of the object. And this would really help, because if we're thinking about it from the quantum circuit and the quantum computing perspective, if we know that there are some symmetries, we can make use of this symmetry so that we have to use less qubits to initialize in our circuit to time evolve, which can overall give us different types of working perspectives of some compounds that we can run more naive quantum circuits on, from which we can build upon and look at more complicated quantum circuits for time evolution. And for the COVID-19 receptor, a symmetry of the receptor structure also plays a significant role in allowing comparisons between classical and quantum methods of computation. And this was from the diagram in the reference that I've been mentioning several times throughout the presentation, in which they looked at 92 possible crystal structures, and they looked at the way in which binding pockets can be distributed along a rotation in this way, a clockwise or a counterclockwise rotation about different axes of the COVID receptor, and how this can relate to quantitative and computational biology, and can also have connections with quantum computing if we can look to determine whether there's some type of quantum encoding of the information that's given in the molecular structure, and how we can make use of this information to run the quantum algorithm in polynomial time for generating some readout, which can then be reconstructed and put together to approximate the binding affinity. And with these lessons in mind from classical methods from several classes of receptors, we are interested in determining amenable qualities of quantum circuits to test for cases of the quantum method. So we just want to be able to determine whether, even if a structure or a compound is too complicated, whether we can isolate some smaller part of the topology of the surface of the receptor, and whether from the restricted topology of the surface of the receptor, whether we can still run the algorithm, which would then give us some encouraging type of result of being able to run a larger portion of the topology of the surface of the receptor on other future near-term quantum hardware. And to work towards this goal, we first observed that quantum circuits introduced from the variational quantum algorithm for time evolution and Lubash et al. Can be appropriated in a very similar way for the electronic structure computations. And this is very much given in a type of iterative optimization loop that was given in the previous workflow, and that we need to think about from the circuit, or from the structure, the molecular structure of the receptor, like how many qubits should our quantum circuit has, and if there is any rotational symmetry of the receptor structure, and can we make quantum circuits more shallow with less qubits to speed up the structure computations for determining properties of the electronic structure, as well as from the fact that from the direction of the quantum circuit to the molecular structure, whether there are any other closely related receptors for new structures that we can study from quantum circuits. So it's just demonstrating that we can make use of the same type of iterative optimization loop for either being able to improve our quantum circuit construction from information of the molecular structure, and likewise being able to determine whether there's any other type of better or more optimal molecular structures from how the quantum computation goes from the time of evolution on the quantum circuit side of things. And for quantum computing, a few different objects have to be introduced from the following reference. So we can think about quantum algorithms for electronic structure calculations, which is very much going to formalize the types of connections that we have been talking about, in which we want to look at and determine whether there are any precise measurements of the binding affinity or other catalytic activity of different types of compounds and receptors and ligands. And we have some publicly available information on it, but there can also exist a Hamiltonian formulation, as we have been mentioning and alluded to in an earlier slide, which can help us formalize these notions and be able to discuss about be able to discuss specific qualities of quantum circuits that we want for the time evolution for molecular docking workflows. And the objects defined in the reference are what we had mentioned in a previous slide, which we can go into in more depth now in light of the discussion that we've been having, which is that we're looking at an electronic structure Hamiltonian, which is given of one 2-point interaction and one 4-point interaction between the annihilation and creation operators. And we can form expressions for the 1-point electron correlator and the 2-point electron correlator, which are two expectation values given from the fact that the 1-point electron correlator is just given by the point I, whereas in the 2-point electron correlator function, we have ij instead of just I, and also kl instead of just j. And these two integrals are relating to the fact that they're taken with respect to either one spatial variable in terms of, in the case of the 1-point electron correlator, or two variables dr1 and dr2 in terms of the 2-point electron correlator. But as with all types of quantum algorithms, we need to have some type of very beneficial initial state and ansatz, which they use by the UCC as d ansatz, which is parameterized in the minimum angle. So we can look to directly implement a lot of the notions for preparing an initial state of the solution through the ansatz from the variational quantum algorithm and the variational quantum eigensolver, and being able to leverage and import this framework for electronic structure calculations. In the formal definition of the operations appearing in the electronic structure of the Hamiltonian are related to the fact that there's a whole creation operator, which is given by the Hermitian conjugate of hat of BI in terms of hat of AI, as well as there is a particle creation operation, as well as a whole annihilation operation and a particle annihilation operation. So this is just the basis that we use to define the electronic structure Hamiltonian, and how it very much relates to either adding in or removing individual components of the atoms, which constitute the molecules that we would like to look at the electronic structure calculation of for some compound of interest. And we can parameterize, that we have been mentioning at the beginning of the time evolution, the ansatz, where the EFHF, which is another energy term that's given in the energy of the Gibbs sampler, is given by the fact that it can be expressed in terms of an expectation value, where we incorporate the electronic structure Hamiltonian, rather than in addition to a Bra-Enquête states for the initial state of the solution at the beginning of the time evolution. And besides the previous quantities that were shown, we incorporate the time evolution and quantum circuits into our discussion of molecular docking workflows to compounds from the CellTalkDB repository and the PDB repository for the COVID-19 receptor. And what's particularly encouraging about these receptors is that some of them exhibit more symmetry than others, which can give us an idea about whether the quantum algorithm could still exhibit some type of competitive performance for the molecular docking in silico experiment, even if a compound does or doesn't have symmetry. So in the more optimistic case, in which some compound, the receptor structure has some type of like rotational symmetry or other types of symmetries, we could try running the quantum circuit for the time evolution on a more shallow quantum circuit, which would require less qubits to initially parameterize and less qubits to time evolve, and also less qubits to generate some type of measurement from, with the hat of the poly Z gates, which we distribute at the last stage of the quantum circuit in the time evolution at each iteration of the optimization loop. So this is just demonstrating how we can pretty much exactly look at different types of connections between the classical algorithm and the quantum algorithm, and how in many cases the quantum algorithm can offer some type of different perspective, which can help us further iterate on enzyme design or different design of compounds, which can interact optimally with some receptor. And this is to say that each receptor raises its own intrinsic challenges, not only through the number of possible sites at which a ligand can interact with a binding pocket, but also through other through other conformations that the receptor can have with different molecular vesicles in the body that can ultimately determine how an infection is initially propagated. And this is to say that as we reiterate, the COVID receptor can be a particularly difficult receptor and tricky receptor to characterize in silico, because there are many different possible crystal structures of how this receptor is, and it can be difficult to determine how to incorporate different characteristics from the crystal structure into training set data. So this is just to reiterate from the reference that we had been mentioning, in which I had examined with another open source molecular docking algorithm. For the COVID-19 receptor, there are 92 possible crystal structures, as well as more than 10 possible binding pockets, and rotating the receptor structure of COVID-19 reveals very different topologies that are not very symmetric within the structure that are associated to each binding pocket. So it's just demonstrating that it can be difficult to try to run a quantum algorithm in one shot or in one initialization step for all of these types of pockets at the same time, in comparison to actually looking at the isolated topologies of different binding receptors or different binding pockets along different components of the receptor. So this is very much something that a researcher needs to keep in mind on a case-by-case basis, depending upon the receptor that's being looked at, which can give an idea about how many combinatorial outcomes there are for interaction between a receptor of interest and the binding pocket from some isolated topology and different types of ligands or different areas of the receptor altogether. And the Hamiltonian for the electronic structure calculations shares in many similarities with the Ising model Hamiltonian from statistical mechanics, with differences in the generation being the annihilation and creation operators. And this is given by the fact that the Ising model Hamiltonian can be taken under boundary conditions eta over some finite volume lambda over the square lattice, and we're looking at the Hamiltonian or the energy function for spin configuration sigma without the presence of boundary conditions or an external field in the Hamiltonian for the Ising model. We can have the expression for the Hamiltonian being given by minus the summation of jxy sigma x sigma y. And having this thermodynamic interpretation and type of mapping or equivalence between an electronic structure Hamiltonian and an Ising model Hamiltonian, it's helpful because it can help us form some types of thermodynamic interpretations about points of an amino acid chain at which the thermodynamic interactions in a binding event are more strong, and they predominate in some isolated regions of the receptor structure in comparison to other regions, as well as allowing for computing 1-point and 2-point electronic correlators relating to those expectation values, which I had exhibited in an earlier slide in the presentation. And as a beginning example of one compound that we can study more closely with the quantum circuit approach, consider the following structure. So we can see that we have produced one homology model from the CellTalkDB receptor, which is given on the left-hand side. And we can see that the characteristics of this molecular structure are related to the fact that it's composed of several helices that are tightly occupied within the region of space between the two circles that are produced in the homology model, as well as the fact that the helices near the top of the compound fold in some non-trivial way, which distributes more weight of the compound away from the bottom 3 helices. So when we're running a molecular docking simulation, we very much have to think about different ways that the interactions could occur, either along the helices in the bottom region, along the bottom region of the compound, or along the top region where the helices fold. And characteristics of the simple molecular structure raise difficulties of quantum circuit encoding, in which we want to think about whether we have some amino acid representation of a compound, whether there can exist some quantum circuit encoding or quantum circuit representation that we could time evolve for generating some type of favorable measurement and readout. And in another presentation that I've discussed on the channel for fast quantum algorithms, which is a different class of quantum algorithms rather than the variational quantum algorithm. The fast quantum algorithm exhibits us that there can exist a polynomial scaling relation or a scaling type of law between the number of qubits that you would require in the circuit versus the number of amino acids which are contained in the compound, which is given in this case by 16n cubed minus 24n squared minus 12n plus 63. And with this scaling relation in mind between the number of amino acids and qubits in mind, even more computational complexity arises for the COVID receptor and its many variants. So it's very much of interest for future and upcoming research to think about how we can make use of these different types of notions between quantum computing and time evolution, and even the number of qubits that we would require on the circuit depending upon the number of amino acids, in which for the COVID receptor, the presence of rotational symmetry makes or the lack of presence of rotational symmetry makes encoding a quantum circuit for the receptor structure costly. So this is to say that in some cases, running a quantum algorithm for generating this type of readout could be very difficult and in fact prohibitive to do, because the quantum circuit to time evolve and even to initialize could be that even if we do scale up the number of qubits that are involved in the circuit, it would really prevent us from being able to generate any type of meaningful output. And to this end, quantum circuit design continues to remain of great interest for investigating novel properties of compounds in the long term, or even in the near term or in the short term, in the long term, however you want to phrase it, in which we want to think about, are there more general features of quantum circuits that can be constructed in Qiskit or in Cirq, like, Cirq is relating to the framework that I've already investigated, but there are many ways in which one can look to also design or implement time evolution on quantum circuits in Qiskit from quantum circuits in Cirq. And also, can such simulations be executed over the cloud with emerging near-term hardware? And also, in the third item, would any difficult gates that are difficult to simulate appear in newer classes of quantum circuits? And would such gates appear more than once throughout the time evolution of each iteration that we're running the quantum circuit for? As well as finally, would there be any speed up with a quantum circuit as the depth of the circuit increases? And so I wanted to end the presentation on some conclusion, which is relating to the fact that we motivated the utility of molecular docking workflows in the fields of enzyme discovery, directed evolution of enzymes, machine learning, and deep learning. And we introduced classical and quantum methods of computation for simulating properties of compounds relating to the electronic structure and other chemical properties. And we discussed how to apply quantum algorithms for the COVID and CellTalkDB receptors. And still, even though we have some initial impressions for what a quantum algorithm for molecular molecular structure prediction or drug discovery or directed evolution of enzymes would entail, there's very much many other types of compounds which would be interesting to examine further in this framework. And we gave an overview to this end of the current prospects and challenges. And thanks for listening.